Show with Jay Patel. Good evening. Tonight on MTP, we're going to talk about cell transport. What is cell transport? Cell transport is the movement of compounds across the membrane or outer walls of the cell. Why is this vital to the cell? This is vital to the cell because it maintains the function by allowing the cell to release and take in compounds. Also, it regulates the characteristics of the transporter. So moving on to a specific type of transport, membrane transport proteins. They transport substances which involve the movement of ions, small molecules, macromolecules across a biological membrane. They are also known as integral membrane proteins that exist across the membrane where they transport substances. These proteins help with the movement of substances by facilitated diffusion or active transport. Tonight we're going to have several speakers that are going to talk about specific types of transport. Give a warm welcome to Nurse A.T. Pope.
Thank you, good to be here. So as Jay said, today I'll be talking to you about pumps, and to further explain that, I will show a video.
Pumps is a class of membrane transport proteins that generate and maintain chemical or electrical gradients. They require energy input in which they have to break some bonds to produce energy. Pumps are very specific and transport ions and small molecules. Sodium-potassium ATPase is expressed in most cells. It catalyzes the exchange of 2 potassium ions for 3 sodium ions. Because of the uneven exchange in addition to ion gradient, the pump produces voltage gradient, which is electrogenic. In this demonstration, a sodium-potassium pump is used to maintain ion gradients. The sodium ion concentration, represented by the blue, is kept low in the cytosol, while the potassium ion concentration, represented by the yellow, is kept high in the cytosol. Sodium ions enter the pump on the cytosolic side. Pumping sodium against this concentration gradient requires energy by using ATP. ATP transfers a phosphate group to the pump by energy linkage. Phosphorylation occurs, which causes a change in the pump's conformation, opening the pump and releasing the sodium ion outside the cell. The potassium ion then goes into the pump. Binding of the potassium ion causes the phosphate group to leave, which then releases the potassium inside the cell.
Thank you for the explanation there.
Thank you for having me.
Next, we're going to have Professor Chan, who's going to talk about ion channels.
Thanks for having me on the show, Jay.
My pleasure.
As Jay said earlier, today I'm going to be talking about ion channels. I actually have a video that my students prepared for me in the lab. Ion channels are water-filled pores in the membrane that work by opening and closing. There are non-gated channels that are always open. These channels are known as leak channels because ions are allowed to pass through them without any trouble. There are also gated channels that are closed until activated by stimulus in order to open. Stimuli can be mechanical, such as a stretch in the membrane, thermal, chemical in the form of ligands, and electrical with the use of voltage. Once pore is open, ions flow from high to low concentration. All ions operate with the existence of a gradient made by a pump, such as the sodium-potassium pump, and no new energy is required. Ion channels are highly selective and can transport sodium, potassium, or calcium. Calcium channels are very important in the initiation of muscle contraction and hormone secretion. Other ion channels, such as the ones involving sodium and potassium, are highly used for creating action potentials in the nervous system, which in turn are responsible for vision, pain sensation and detection, memory, signaling, and many other important functions. This is an example of a voltage-gated ion channel. This channel starts off closed until it is opened by the sensation of the voltage stimulus. Then an ion on the right side is transported down its concentration gradient. Ions are transported until an equilibrium is reached, then the gate closes until it is disturbed by the stimulus again.
Thank you for that demonstration, Professor Chan.
Thank you, Jay. It was wonderful to be on your show today.
Next, we're going to have a student from UCLA, C-Tran, to talk about cotransporters.
talking to you about co-transporters. First, let's see a demonstration of how importers work.
Co-transporters use gradients created by pumps to transport other molecules. They require the existence of a gradient of one molecule. Energy accumulated in the concentration gradient allows for the transport of a second substrate against the gradient. Co-transporters undergo conformational change. Transport is coupled. Neither molecule can go without the other, and different number of molecules can be transported in different directions. The two kinds of co-transporters, symport and antiport, transport one substance down the gradient and use this motion to transport another against the gradient. Let's start with symport. Symport is important for transport of nutrients through the epithelium. In this demonstration, the coupled transporter moves solutes in the same direction across the membrane. The green solute is carried down the concentration gradient from high concentration to low concentration. The energy released by the movement of solute drives the movement of the other solute, which causes the orange solute to be transported against this concentration gradient from low concentration to high concentration.
Interesting.
Well, now let's take a look at how antiport works.
Antiport, passage of the molecule against the gradient regenerates the preferred conformational state. In this demonstration, the coupled transporter moves solutes in opposite direction across the membrane. The orange solute is transported down the concentration gradient from high concentration to low concentration, fueling the transport of the other solute. The green solute is then transported against this concentration gradient from low concentration to high concentration.
Wow, those were some very informational clips. Thank you for being on the show today, Sabrin.
No problem. I'd love to see you chat, but I have to get to my research lab.
Next, give a warm welcome to Coach Glucagon, who's going to talk to you about uniporters.
Oh, so glad to finally be here, Jay.
Glad you could make it. So about uniporters.
Well, but uniporters are relatively quite simple, actually. I have a short video for you guys.
Uniporters are intrinsic membrane proteins that transport substances down their concentration gradient until equilibrium is reached. It is highly selective for the type of protein it transports, and it is also reversible based off of the concentration. Transport through a uniporter is also very saturable in that if there are only a low amount of transporters and a lot of substrate, the transporters can become clogged up. In this case, since the transporters only transport one substance at a time, the molecules would be required to wait in line until it is their turn to be transported. One very important example of this type of transport involves the glucose transporters, which are called GLUTs for short. There are 13 different types of GLUT transporters that are expressed in different tissues in the body. This example shows two uniporters. On the left side, the concentration of molecules is expressed at a very high rate. Since the transport occurs from high concentration to low concentration, the molecules from the left side will go down their concentration gradients to the right side. Once this process is over, the molecules will be at equilibrium, which is 4 on the left side and 4 on the right side. This process would repeat when there's another concentration imbalance.
Thank you for making that so simple for us, Coach.
Well, thanks for having me on the show. We'll actually have a game to win, so I'll catch you later.
Now we're down to our last speaker, Librarian Fusion, who's going to come out and talk to you about diffusion. Librarian Fusion?
Yes?
Aren't you supposed to talk to us about diffusion?
Oh! Well, Jay, I could actually talk to you about diffusion, or I could read you a paragraph about diffusion, but knowing the newer generation, you'd rather hear about diffusion. Here's a short clip for you.
Diffusion is a type of transport that does not rely on the help of membrane transport proteins to operate. In this type of transport, the imperfections in the membrane bilayer and laws of thermodynamics facilitate diffusion. It is a very simple type of transport, but keep in mind that not all molecules can be transported in this way. Only small uncharged hydrophobic molecules can interact with the bilayer and be transported in this way. Two important examples of molecules that undergo diffusion are oxygen and carbon dioxide molecules. This example highlights the fact that there are no transport proteins involved in this type of transport. Molecules move from high to low concentration until an equilibrium is reached. Once there is another concentration imbalance, the procedure repeats until equilibrium.
Well, that was very insightful.
Oh, thank you.
Thanks to all the speakers that came tonight. I definitely enjoyed listening to all the insightful conversations you held. I think we can all say that we learned a lot about membrane transport proteins. Until next time, this is Jay Patel on MTP.